is going to be a great talk this morning. So my name is Brent Edwards. I'm from Calgary, and I'm very excited to introduce today's speaker, who is Marjolein Meulen. And Dr. Meulen is the James M. and Marcia McCormick Director of Biomedical Engineering and Swanson Professor of Biomedical Engineering in the Menig School of Biomedical Engineering and Sibley School of Mechanical and Aerospace Engineering at Cornell University. She's also a senior scientist in the research division of the Hospital for Special Surgery. She has many accolades. She's the recipient of an NIH First Award, an NSF Faculty Early Career Development Award, and she has been deputy editor for the Journal of Orthopedic Research, a member of the ASBMR task force on AFF, and is a member of the Skeletal Biology Structure and Regeneration Study section on NIH. What is not mentioned in your program is her dedication to training and mentorship, and I first met her at an MSK meeting in Sun Valley, and I will always be very thankful for the advice and guidance that she provided me as a young postdoc transitioning into my first faculty position. So I thank you for that, and in the interest of time, let's please welcome Dr. Marjolein Meulen.
So thank you, Brent, and thank you, the organizers. Art Kuo is a friend of mine, one of the first people I met in graduate school, and so it's nice to be invited and see Art on his home turf. So I'm going to talk about the adaptation, loading, mechanobiology of musculoskeletal tissues, and I want to talk what I'm calling it the yin and the yang, the good and the bad. We're doing a lot of this work, and I want to show you the story of where this can be beneficial and where it can be detrimental. So I want to acknowledge my disclosures. And so I, like many of you, am interested in musculoskeletal function, and I started as a pure bone person, interested in how the skeleton supports loads, but more importantly, that these days to me is not just the function, but it's actually what the function does is it creates forces, and these forces adapt the skeleton, and they adapt the tissue structure, and they affect the cells and the biology. And so this adaptive process is really what my lab is interested in, and what we're looking at in musculoskeletal diseases. And you can really take these loads and think of them in multiple categories. So you can think of things that increase loading, things that decrease loading, and then situations where loading is altered. And I think all of these three situations give us great insights and provide us opportunities to understand this process of the response to mechanical stimuli. And then what you then get are changes in the tissue, be that tissue, bone, tendon, cartilage, in terms of composition, just how much tissue you have, and also then the structure of that tissue. So this is where this is where we focus on in my lab, and this is what I'm going to talk about today. And really what this process is a process of mechanotransduction, from putting a stimulus onto whatever the structure you have, looking at the pathways, and the arrow is deceptively small because there's many pathways involved, to then understand the cellular response and the tissue response. And I think that this response is what we probably know the best, there's a lot in this little arrow and in the mechanical stimulus that still needs to be understood. And so what I'm interested in is understanding the mechanisms of this adaptive process, and really ultimately we want to use this for potential therapeutic approaches and treatments, and understanding the efficacy of therapeutic approaches as well. So when we think of the skeleton, there's two types of bone tissue structurally. There's cortical bone and there's cancellous bone. And when we look at understanding this response, I think you have to examine these two separately, because they have distinct responses and they're not always the same. And if we think about cortical bone, there's most of what we understand in the literature is actually around cortical bone in terms of mechanotransduction. And I think the tools and the various models for looking at this are pretty well developed, and they're really the basis for most of what we consider how adaptation to mechanical loading works, in fact. But then on the other side, cancellous bone, it's, I think, more clinically relevant. As I've shown here, osteoporotic fractures occur at primarily cortical cancellous sites, and implant osseointegration is very much determined at cortical cancellous sites. But when we started thinking about these questions now some time ago, there were very few in vivo models developed to understand them. And in particular, cancellous bone is really very much often affected documented in various mouse genetic models as well. So, and again, in mouse genetic models, the response of cortical and cancellous bone is different, and so we really wanted to be able to modulate this tissue and understand its loading response. So that led us to develop a model of in vivo compression of the mouse tibia that then allows us in this tibia, so let's see if this little cursor, so we have here the tibia that's cupped at the knee and held at the foot. Is that, yeah, it's showing up. And then you now not only can look at the cortical response to the mid-diaphysis, but you now can also look at the cancellous response at the proximal metaphysis. And in our hands, we use a small handheld device that's instrumented, pretty simple to use. And then I've shown the waveform for everything I'm going to talk about today, where we use we only apply 5 minutes a day of loading, so which is it's at 1,200 cycles at 4 hertz. It's got a dwell in there, which because we're trying to achieve this 4% strain rate that's been shown to be the rate at which the mouse more or less ambulates and loads its skeleton. And then we load 5 days a week with 2 days off for the weekend. And then the contralateral limb is our control, and we've shown in some experiments that our contralateral response is similar to a sham response with a sham unloaded animal, so that we use that limb, and that's not always the case for everybody, but we don't have any systemic effects in terms of the tissue level response. So when we use this approach to loading, we find an anabolic effect in bone. And so shown here is a micro-CT scan showing male and female data. The top is the female, then the bottom is the male data with the loaded limb, and the control limb is just a slice taken out of the proximal tibia. And then what you can see in the data here for bone volume fraction, the male animals have more bone volume fraction to begin with, but the magnitude of the response is similar in male and female animals to loading. And what we have is an increase in bone mass occurring. And then I'm not showing the full data set here, but this response is actually percentage-wise greater in the cancellous bone than in the mid-diaphyseal cortical bone. So that's where we've moved forward with this as a good model for looking at the cancellous bone, but we continue to look at both sites. And in fact, you can also look at the cortical bone in the metaphysis as well, the cortical shell. And then in our early work, we don't do this all the time, but in early work, we also developed some finite element models from the micro-CT scans to show that this increased bone actually increased the stiffness of the metaphysis, and it also increased the load sharing by the cancellous bone versus the cortical shell. And so we have an increased stiffness here in this model as well in the proximal tibia. So we then, I presented some of this work to some collaborators of mine and said, we have this great model, we can induce bone formation. We've shown in some other that we can inhibit the loss of bone mass. So if you do orchidectomy in male mice or ovarectomy in female mice, which induces bone loss, you can load and actually maintain bone mass or maintain and or increase. I'm not sure, it depends on a little bit on the mechanism there. And then we've also spent a lot of time, because of our interest in postmenopausal osteoporosis, looking at the response to estrogen and whether that's altered, and in fact, the response to loading is not. But what we're showing then is this proximal metaphysis is stiffening, and then one of our collaborators at Special Surgery, Steve and Mary Goldring, our two collaborators, made this point that, in fact, we're loading through the joint. And as someone who was solely focused on bone for the early part of my career, I hadn't thought much about the joint tissues. My goal was to increase bone mass. However, this metaphyseal stiffening is not happening in isolation. And so it let us step back and say, what is the systemic musculoskeletal response here? So we, thinking of it as only the skeleton is a little bit limiting. And in fact, we have this osteochondral composite, so we're transferring load through the joint from the cartilage to the bone, and there's meniscus in there. There's also within the cartilage, calcified cartilage, so there's a whole structure in there that's also being loaded in this process. And presumably, this stiffening of the this bone substrate underneath these tissues, these very soft tissues, may well be altering their stress state as well. So that led us then to think about the joint. And this is where our collaboration with the Goldring started. And I just want to make a couple points about the process of OA in the initiation and progression. This is a chart. It's a little busy. Look, I'm going to focus on the top half of it, showing the progression from initiation to early development of osteoarthritis to the further progression of it and to late stage. And most of what we see in human x-rays is, in fact, late stage osteoarthritis. But if we look at The top two boxes in terms of what's contributing to both the initiation and the early onset of osteoarthritis, we can see that mechanical injury is listed as one of the initiating factors, which is post-traumatic OA, PTOA, and then also repetitive injury and subchondral bone changes. And so we said, well, are we creating subchondral bone changes, number one, and number two, what is happening at the joint? So this, and then there's a lot of literature, so this led us to think about the joints and go to the literature, and as was presented in one of the keynotes yesterday, there's a lot of literature on osteoarthritis and mechanical loading, one of which right now, which is receiving a lot of attention, is the fact that obesity is a major risk for OA, and it's a little more complex than just loading, I think, but nonetheless, it's very much associated with OA. Occupations with heavy lifting increase susceptibility to osteoarthritis, and moderate physical activity and weight loss both are known factors for reducing symptoms of osteoarthritis. So there's a lot of correspondence between these two, between loading and the development of osteoarthritis, and development and progression. It's hard to separate those two out, in fact, but I do think they're distinct events or processes. So that led us to ask the same question in cartilage and the joint as we had been asking in bone for much of my career, which is, how does a mechanical stimulus lead to a cellular response? And in particular now, does our loading model result in a cellular response at the joint? And so then we started looking at this question, and I just want to sort of orient you because I'm going to show you some images. So first of all, most of our bone work, which I've only showed you a little bit of, is in female mice because of our interest in osteoporosis, but estrogen is chondroprotective, so we've done a lot of our loading for looking at the cartilage work, at the osteoarthritis work in male mice. We're doing this in 26-week-old adult mice, and it's the same waveform as I showed you, but I'm going to show you two different load magnitudes. And what I showed you earlier was a 9 Newton peak load, and now we're also going to look at half that here, which is a 4.5 Newton load. And then I've shown here a section through the knee. We focus on the tibia, so that's the bottom half, the tibial plateau. So this is the tibia right here. The femur is for sectioning, for embedding, is extended out of its normal flex position, and so that cartilage is not the loaded cartilage, so we don't look at that plateau. And so then I'm going to show you some images showing the tibial plateau, and in these images, red is cartilage. It's stained with saffron and O staining, which makes a beautiful red stain of the glycosaminoglycans in the matrix. So the intensity of staining, it gives you an idea of matrix composition, and then the blue substrate underneath is the bone. So we started off with a simple experiment looking at adult animals with 3 different time points of loading, and then looked at what was happening in the cartilage. So shown here are these images. So the first column is the control. So the top row is the low load, the 4.5 Newtons. The bottom row is the high load, 9 Newtons. The first column is control, and then the next 3 columns are 1, 2, and 6 weeks of loading. And so again, the red is the cartilage, the blue-green is the bone, and I think you can appreciate looking at the cartilage that, especially on the 9 Newton load, that there is damage happening on both on the surface of the cartilage. There's actually frank loss of tissue, and also the intensity of staining is altering with loading. And then we score these, which basically verifies what I just showed you, but quantifies it. So shown here, the green is the 4.5 Newton load, the blue is the 9 Newton load level, so twice as high, and then showing you, and the yellow is the control limb, again contralateral. So we see then is that with loading, we are increasing, we are creating damage, we're increasing the damage score from histology, from the Orsi scoring system. There's an effective loading, and that effective loading then is different by load level, so it's more severe at the high load level, and then it's also increasing and becoming greater with the load duration. So then the other thing, if you zoom out on our joints, we looked at osteophyte formation because this was really, really obvious in this model. And so shown again on the top, it's the same layout, are the low load. And if you look at the joint margin, so the side of the joint, the edge right here in the control and in all the low load animals, it's a nice clean joint margin. Again, where this is zoomed out a little bit, so you're seeing the articular surface of the tibia as well as the growth plate here. Now if we look down at our 9.5 Newton animals, what we see is a massive amount of cartilage developing and then mineralizing. So this is an osteophyte formation, and in our high load animals, it's present in every single animal, and it's not there in the low load. And it's something that's in the various conditions. Sometimes it's not there 100%, but it's often at the high load it really is. So what we have then with loading is that we recapitulate osteoarthritis formation. I didn't show you all the subchondral bone changes, but we have over time at the 6-week time point an increase in bone mass. We have at the subchondral bone plate, we have degeneration of the cartilage, which I just showed you histologically, and also with the scoring, and osteophyte formation happening at the margins of the joint. So what we have now is a beautiful model of load-induced osteoarthritis, and I want to sort of emphasize that. And this is a figure from a paper from a couple years ago that talked about the various phenotypes of osteoarthritis, and I've added the yellow load-induced because I'm not quite sure where in the schematic that was presented in the manuscript in that paper to put it, but I think that all osteoarthritis is not equal, and I don't think we know enough to distinguish one from the other, and it may be that this load-induced phenotype is actually a subchondral bone phenotype because of the responsiveness of the bone, but nonetheless, I think it's a particular phenotype and a particular model of osteoarthritis. So then what this model gives us an opportunity to do is to really drill down a little bit in a disease where we have a lot of unknowns and don't understand all the changes. And so I think most human OA is combining some of these factors, in fact, and it's spontaneous, but I think that everything we see from the clinic is very, very late stage. We don't know enough to look at early stage, and so these animal models provide an opportunity to look at this pathophysiology and to understand, and I think different models match different aspects of the human disease, but nonetheless, they give insight into that process. So now I want to go back to this model that I just showed you our data on, and so we are interested, because we're interested in the role of the loading in this process, we then said, well, what are we doing loading-wise? I want to understand how loading is developing this damage in these tissues, but what is the loading we're applying, in fact? So we did a small study looking at to characterize the joint kinematics with loading. So we did an RSA analysis with a modified single plane. We did just did two planes in sequence. We loaded up to the 9 newtons. We did it stepwise and collected data. And then we also did continuous to validate that the motions are the same in the stepwise situation as in the continuous load situation. And so these are 2 videos of the 2 orthogonal planes. I've outlined where the bone sits. I'm going to remove the bone so you can just see the compression happening of the tibia. There's some rigid body motion happening, and there's some deformation happening. And really everything is happening in one plane, so I'm going to just show you that blown up a little bit, so you can appreciate the motion that's happening. And so there's obviously 6 degrees of freedom that we can have on the tibia. And then if we look at it, we find we really only have 2 translations happening here, ultimately, when you process the data. And then it turns out that we have some anterior motion of the tibia happening, and then the compression, the axial compression that we expected and thought we were inducing, indeed, intended to induce. When we look at the femoral motions, we find that there's a rotation happening of the femur that's in this plane, and that's actually contributing to shear at the joint. So what we have ultimately really at the knee with this load model is a certain amount of AP motion, which is creating shear at the surface of the joint. There's rotation of the femur, which is further adding shear to the joint, and there's the compression that we intend to apply. And we have no out-of-plane motion, so which is in the medial lateral directions. It's all anterior-posterior motion happening here. So when we did a series of trials of animals of this, find that it's a very consistent and controlled motion across trials and across animals. The damage in the cartilage then has to be due to either this compression that we intend to apply or this AP shear that we're inducing. The osteophytes that we see are located medial lateral. Actually, they're medial primarily. And we thought this was associated with medial lateral motion because if you look at the human literature, everybody talks about how osteophytes help to increase the joint surface. And so we thought this was, again, medial lateral activity happening, but it turns out it's not that because we really don't measure any medial lateral motion with this model. So then again, so now we know something about the loads we're applying, and now we wanted to drill down and understand further what's going on at the tissue level and in the stresses and strains that are being experienced. Because of resolution issues in the mouse, we've done some modeling, so I'm not going to talk about that. So we can do finite element models to look at the stresses and strains in the tissue, but we've been interested in looking at this experimentally as well. So what we've done is taken an approach to modulate the properties and look at how that changes the response. So we can either alter the properties of the subchondral bone, or we can modulate the cartilage matrix and look at what's happening there. I know I just realized I modified the next slide, but not this one. So this is work that we've done in our lab, but then in terms of looking at the modulating the cartilage matrix, Andy Pizzolitti's lab has also been doing this, who's also been working a lot in this area. So I'm going to show you a little bit of data. We've published something on modulating the cartilage matrix, and then interestingly, both the data from our lab and the data from the Pizzolitti's lab that use genetically induced OA models, so animals that just spontaneously develop osteoarthritis over time. We did not, neither of us, and they're very different models, saw interactions between the load-induced form and the genetic form. So that's interesting, although I don't think it says that's never the case, but they are, again, going to this multiple phenotypes of OA situation. But I want to show you some data we have for altering the subchondral bone and how that affects the damaged development, really. So there's been a lot of work done on the role of bone mass in OA, but a lot of older work, I would say. But we were interested because we came at it from looking at estrogen deficiency, where we're looking at this in bone with load and menopause. And menopause and overectomy in animals are both associated with increased OA development and severity. And there's been a big question, as I referred to earlier, about the direct effects of estrogen on joint tissues, which is why most of the work in cartilage gets done in male mice. But then we've been working, like I said, in female mice to understand the role of estrogen loss. And estrogen is associated with increased cartilage turnover as well as decreased bone mass, which is what we associate with postmenopausal osteoporosis. And also in the subchondral bone, there's also decreased bone mass. So we wondered whether this load-induced osteoarthritis then, what the role of the bone properties is in this process. And so if you lose bone or if you increase bone mass, how does it change the development of, does it change the development of damage? And we've been coming at this question several different ways, but this is a model that we had to alter the bone properties genetically and look at what was happening. So we hypothesized that we would attenuate the load-induced progression by lower subchondral bone mass and stiffness. And so if you look at the literature, there's a lot of discussion around increased stiffness of bone, increases the stresses on cartilage, and therefore increases osteoarthritis severity. So what we used is a model here. It's a conditional knockout model. It's an osteoblast-specific conditional knockout. So it knocks out estrogen receptor alpha. So estrogen signaling, it's one of the receptors for estrogen signaling only in osteoblasts, mature osteoblasts. It's with the osteocalcin promoter. So these animals are missing estrogen signaling through estrogen receptor alpha only in the bone. And in bone that's mature bone, so it's either, it's in osteoblasts and in osteocytes as well. So this is a bone-specific estrogen loss, and so it's only, in the conditional knockouts, they're only missing estrogen in bone, so their estrogen in cartilage is there and is fine and is still signaling. So first of all, we demonstrated that we have reduced bone mass in these animals, and this is looking at the 3 different sites. And so the first one is the subchondral bone thickness, that you can see that there's a loss there. There's more of a loss in the metaphyseal bone, but nonetheless, the whole proximal tibia should be less stiff. There's less bone there. Then we also validated that there's no cartilage phenotype here, and so the cartilage thickness, and also when we do the Mankin score, which is a structural assessment of the cartilage, is the same between the, it's not different between, and if anything, the conditional knockout is on the leaning towards having more pristine cartilage, so to speak. And then we loaded these animals, and for a variety of reasons, we used 3 different load levels associated with a different part of this experiment. So I'm showing you 6, 7, and 9 newtons, 6.5, 7, and 9 newtons of loading. And so again, I'm showing you the histology, and now I have a column of the conditional knockout, the conditional knockout here, and the littermate control with the control limb on the top, and then showing the various load levels after 2 weeks of loading here. And if you look at the change in the mean Orsi score, so the delta between the 2 limbs, what you see is A, a beautiful dose response to loading, and B, that all the red bars, the mean of all the red bars is higher at each load level than the littermate controls. So we have more cartilage damage happening here in a situation where there is still estrogen signaling in bone, but that we just have less bone there, less substrate underneath in the subchondral bone and in the epiphysis and metaphysis. Then we also looked at the osteophytes in this model, and we find that we have larger osteophytes with loading and in the conditional knockout animal, again showing you a column of the littermate controls and a column of the conditional knockouts, and with the osteophytes circled here where the osteophytes are forming on the joint margin. And so again, there's a load-based dependency in that they're more mature, so there's more cartilage in the osteophyte at the higher load levels, but nonetheless, even in the lower load levels, there's a pre-osteophyte forming as well. So what we have here then is a high magnitude in vivo loading, that is inducing an OA-like pathology in the mouse joint. I've shown you this in several models. And then we've shown now that with reduced subchondral bone mass and associated with that the stiffness, although we haven't tested stiffness directly here, that we increase the damage that happens with load-induced osteoarthritis formation. So it's really, so we've shown very well that we can damage cartilage and we can damage it very consistently. However, we spent a long time discussing, could we do something beneficial for cartilage? And so could we use the other end of the spectrum to try to actually attenuate, again, I'd love to attenuate, not just increase the damage of osteoarthritis, of load-induced OA. And there's data showing that treadmill exercise can inhibit damage after ACL injury. And for those of you that don't know, ACL injury is a commonly used model of osteoarthritis in vivo. So, we spent a long time talking about how does one design an experiment, because it's also potential at a low load to show no effect. So how do you get an effect versus a zero, just nothing? So we spent a long time on this, and ultimately decided to do an experiment with surgically induced osteoarthritis through the DMM model, the destabilization of the medial meniscus model. So we surgically created OA here, and then we applied load. So we first, so this is now in male mice, in contrast to the previous study. So male mice, and these are actually young mice, which we previously showed have similar cartilage damage as the older animals. So we did the surgery, and then 5 days later, we started looking at the animals. And so we have just a 5-day time zero control, and then we looked at 2 and 6 weeks further. Sorry, this is animated differently than I expected. And then we applied loading, and we applied a low load level. So what I showed you earlier was a 9 Newton. Here we applied a 1 Newton load level at this 5 days after the induction of the surgery, of the starting the damage in the cartilage. And then we looked at the loading at 2 and 6 weeks post-loading. So I'm going to show you the data in the same layout, where there's a top row of the control limbs, then there's the DMM only group at 0, 2, and 6 weeks, where 0 is actually 4 days after surgery, and then I'm going to show you 2 and 6 weeks of applied loading. And so what we find here then, and I've shown everything together here sort of in the interest of time, is that we have our control limb, then we have, like I said, the DMM only limb. If you look across that at 2 and 6 weeks, there's very obvious frank damage there, both on the surface fibrillation, and then there's gross tissue loss happening on the surface of the cartilage. And then if we look at so this is now the post-traumatic OA model, surgical model with the 1 Newton loading, what we find is actually that there's a maintenance of the surface, so we don't have the same surface damage. There is some loss of staining happening, however, but we don't have this frank loss of tissue. And then here's the histological score showing this. And so what we then find, again, starting from the baseline data, that at 5 days, there's already some amount of damage there, although they're not significantly different, which we're calling time zero, which is not, but the controls in the DMM are not different yet. If you look at the 2-week time point, then we see that the DMM animals have damage here that's significantly different from our both the control limbs and then our loaded limb, although I guess statistically they are both Bs. And then, but at 6 weeks is where the A group distinguishes the DMM surgery group distinguishes itself from both the contralateral controls and the 1 Newton loaded animals here. So we can inhibit the loss, and we are maintaining the cartilage, both the surface and the proteoglycan loss, the tissue loss, and then the altered chemistry of the composition of the tissue with this loading. And then if you look at osteophytes, we have a very similar result here, where again, this is the control limbs, this is the DMM alone, so this is just the post-traumatic osteoarthritis, where we see that we're getting osteophyte formation. And then if we look at our DMM plus the 1 Newton loading, there may be something forming, but we don't really have anything scoreable, with the exception of one animal here. So they're not. So the DMM is significantly different from its controls. We're not quite different from with the loading statistically, although we are at the 2 weeks, but at the 6 weeks, there's some overlap because of one outlier animal. But there's still very, very encouraging that there's something positive here in these animals. So What I hope I've shown you then, I think what many of us know, in fact, is that musculoskeletal tissues adapt their properties to loading and to habitual loading, and that we can modulate this process, and that this process can be anabolic and can be catabolic. And in bone, we went into this because in bone we knew it could be very anabolic, but in cartilage now we've shown both ends of the spectrum, and I think it's both context and tissue specific in terms of what you're seeing. And I think it's really important to think about this adaptive process as a systemic thing, and the tissue crosstalk between, at a minimum, cartilage and bone, and perhaps more than that, contributes substantially to disease. And osteoarthritis really is a whole joint disease, in fact. And I'm not sure if this crosstalk now, how much of this crosstalk is chemical versus physical. And so I'd like to think that a lot of it's a mechanical mechanism. As a mechanical engineer, that appeals to me. But I'm sure there's been a lot of data recently on signaling across the calcified cartilage and signaling between bone and cartilage. So it may be that the stiffness of the bone is inducing signaling as another way other than just the physical interactions. So I want to thank the organizers for the opportunity to talk today. I want to thank my group. I've shown data for bone and cartilage here from graduate students and postdocs that have worked in my lab. And I also, of course, want to acknowledge funding for this. And I'm very happy to answer some questions if you have any. Thank you.
Okay, great. So we've got a microphone over here for anybody that has a question for Dr. Van der Meulen. Get up, Tor.
Yeah, okay. Tor Bazier, Auckland Bioengineering Institute. Thanks for the really nice overview.
Really nice talk yesterday.
You mentioned signaling, and it's my understanding too, there's also signaling pathways between muscle and bone.
Yes.
So then this thing becomes even more complicated.
Yes.
Although
let me just say one thing, that our loading is under anesthesia, and so I think there may be less muscle contribution directly, but I'm not positive about that.
Right, and I was going to just kind of add there, muscles aren't really a part of this, right? And you've kind of, you have to tease something out to make that consistent. But when you try and put that kind of back into the equation, or think about the habitual loading where the joint's going through large ranges of motion and other things, you've got a pretty aggressive model here is what I understand.
Yes.
And then taking that out back into context, which is what you've mentioned at the end, the context is important. How do you think you might move forward with the current model that you've got to try and explore some of these other areas?
So let me just elaborate what you just said. So I do think it's aggressive, but that's what the model is for, right? I guess if I did this in the mouse and I had to wait 2 years to develop OA, few of us would be using it, right? Sure. So I do agree that it's an aggressive model in that sense, and it's probably potentially more aggressive than we realize. I think there's we are exploring a bunch of things. So the stuff I showed you on the ER alpha knockout was sort of a fortuitous. We weren't really planning on looking at that animal, but we had all the tissues sectioned. And so we've been looking at modulating the bone properties. The nice thing about bone and osteoporosis, in contrast to osteoarthritis, is that there are drugs out there, right? So you can increase bone mass. We tried to do this with 2 different mouse strains that were known to have different subchondral bone properties, just bone mass. But there's a lot of other complexities around the strain-based differences. So we've been looking at changing the bone properties pharmacologically and then seeing what happens as a way of modulating. How much can we modulate this disease with changing the bone? So if you post-ACL rupture, could you give someone a bisphosphonate or PTH, which are both treatments for loss of bone mass, and thereby alter the progression of this disease over time? So we literally have data collected on that right now and are looking at that question at the moment. I also think that I have not talked here, and we're starting to look a lot more at the synovial aspect of the disease also. So I agree that muscle is part of it, but I also think that inflammation is part of it, and we haven't, and I know about this much about inflammation and immunity, and so we're starting to learn more about, collaborate on looking at those questions, as are a lot of other people, I think, as well. So I think for me, there's a question of whether this is bone to cartilage signaling or whether it's bone to inflammation to cartilage signaling also, right? It can be various ways that you can do that. And again, it could also have I agree the muscle is likely in there as well. And I think in fracture healing, we know in bone how important muscle is also. And so I have no doubt the muscle plays a role. But to some extent, this is, if you look at the exercise data, this allowed us to pull out the loading component of that, because exercise induces many other things, and we know it's beneficial in ACL, but then what was driving driving it, and we now know that loading can be part of it, right? So I guess that's how I view this as a question. Does that answer what you are asking?
Sure, yeah. I mean, there's lots to untangle there, but
Yes, yes.
it is an interesting model that you have. So.
Yeah, and again, and I agree, it's a mouse, right? And I would much rather work in something larger and with more tissue to work with and stuff, but at the same time, we can do these genetic manipulations. We're looking at gene expression right now also in these animals, and so that is also a question we can't do in larger animals, and so that's one of the other pathways we're going right now. We're doing a lot of gene expression in the bone, and I think actually in Ralph's talk tomorrow, he'll address some of that as well in the afternoon talk tomorrow.
Thank you.
Speaking of me, right?
Dr. Meulen, how are you?
Ralph Meulen, ETH Zurich. Marjolein, very, very nice data. I would have asked you about immune responses, but you already said that you don't know much about it yet. I think
I know a little bit, actually, but I don't know how much I want to say in a public
Exactly.
talk. But we are starting to look at it.
I don't know much about it.
It has to be part of it. I think that's
Yes.
what's got to distinguish the positive from the negative, right? And from what little I know, it's maybe it's the only cells that it's the macrophages are in the joint, but we haven't looked at that yet at all.
Yeah, any kind of T-cell type of
Yes.
that you look at specifically, could you get that out of your tissues if we're looking specifically in these cells? I mean, you have looked at...
So there's no T cells in the tissue initially, but we can look at the lymph nodes, and we have been starting to collect lymph nodes, and we're seeing some T cell changes there, actually.
Right,
so, yeah, there is definitely something happening. So there's an immune response happening, without a doubt. But it's too early. I can't say what it is at this point.
So one of the things that we trying to do is to look very regionally, right? And so
Yes.
when you see this beautiful response to loading, and then it's mechanobiology, but this is not necessarily locally driven, right? You will always have loading and unloading with your models. Did you look specifically where things are happening, maybe calculating local strains and stresses and things like that?
Yeah, so we have. So the damage is posterior because that's where so actually we score the cartilage anterior and posterior. Actually, we've done 6 quadrants. If you go look at our original work, we haven't done that in everything, but we look at medial, lateral, and then within each one, an anterior, a middle, and a posterior quadrant. And the damage is localized posterior. And to go back to Tor's initial point about the range of motion, I think actually this model is limited by the fact that we don't have range of motion in it. So we're loading, I think some of what we're seeing is artifactual because we're loading at a fixed position, and you're not fully unloading because you have to keep a preload or else because of singularities in your load cells, right? So I think there's something there in this response that's associated with the lack of this range of motion that we're inducing here as well. So we are looking regionally. We've also, in the bone side, so I have a whole program going on looking at bone and loading, and in bone, we've been looking at gene expression locally. So we've actually now segmented the cortex and looked at different locations, and our gene expression, the mid-diaphysis versus the next section above it is different. I think we're going to have to go to single-cell RNA-seq for some of that. We're actually looking at tension versus compression now and the responses locally, but I can't do that with the gene expression yet. So I would love to go in that direction, but bone is challenging there, and I think we have to go in that direction to understand what's going on. But in terms of in the bone right now, we've looked at cortical tissue at 3 different locations and are seeing differences in gene expression that we're trying to understand. And again, I've gone at it from a load-based hypothesis that I have the highest load with bending in the mid-diaphysis, and then it's going to get lower. And the gene expression profile is sort of that some of the processes are highest mid-diaphysis, and they turn on at the other locations later. But I don't know. I can't say that it's only a loading-based phenomenon. There may be other differences that are driving that also because there's some tissue-based differences across those locations as well. But we are very much looking at that, the specific.
So one of the problems that we see with cortical bone is that, of course, in your kinds of micro-CT data, our micro-CT data, you don't see local porosity in the cortex.
Yeah.
And that actually will drive different mechanical responses.
Yes.
And it's just like, I mean, the local cells will not see just bending or, or tension on one side, and they will see local changes as well.
Yes, no, I understand.
And how can we, do you have any idea how we could address that with the technologies that we have, or what is needed to actually get a better understanding of the mechanics in cortical bone?
Well, I assume you're going to talk about some of this tomorrow in your talk.
We don't do cortical bone. It's too difficult. So.
Well, but you're going to address it with the cancellous bone, and I'd say we do both. I think I guess as an engineer in this field, the biologists in bone have always wanted things to be random and sample everywhere and show the same effect. I think these are very localized effects, and part of it is, like I said, for me, partly load is what's driving some of it, and I know the loading is different point to point. So, but I agree the milieu is also. So I think think, like I said, on the genetic side, things like single cell, I think the modeling, there's lovely, exquisite modeling being done here. I started in the modeling business, but I got, as a graduate student, but then we got frustrated with the lack of data. And if you go look at my fellow graduate students from my research group, most of us are doing experiments now because we all wanted data for our models. So, you know, I think you need the interplay of both of them. And we have done some of both, in fact, to look at this. So, yeah.
David Lloyd, Griffith University. Sort of following up on Tor's and Ralph's conversation about is it actually a physiological injury mechanism? Because when I get an ACL injury, it's a single insult, and this is a repeat insult, which is getting high strain. So, and this interaction between the bone when you get stiffening, but there's also an interaction when it gets softer, and the cartilage function gets worse when the bone is softer. And that's,
I think that's what we showed in our...
And that's the other side of the coin. Yeah. And there's a lot of work now going on disuse and unloading and all that. I still agree there's overloading, unloading, but I'm just wondering where this fits in the physiological range of what's causing disease. And with your, you know, you said something about inflammation. You've probably got inflammatory effects happening right now because that is the normal course of cleaning up all the debris, getting rid of it, laying down and all that. So we do not have a classic synovitis response, by the way. So we tried to score, we don't have synovitis in the capsule. Okay. We have a fibrosis happening, but it's not a synovitis. So there's not this thickening of this synovial layer. So I didn't show you that data because it's not there actually. So the other question I was really quite interested in is that bone strain to get anabolic effects is actually magnitude and frequency based, so you can have very different magnitudes with different frequencies.
Yes.
And is there a frequency loading or so I won't go loading, I'll go strain, bone strain and cartilage strain that is common, that is anabolic for both?
I think that's an excellent question. So, but actually in these low load experiments, we don't get a bone response because the 1 Newton load we're applying is so low.
But yours is 4 hertz, is it?
We're at 4 hertz, yes.
4 hertz.
Yeah, so we've looked at that. We've characterized all the bone and cartilage in these experiments, and in the low load, we don't have a bone effect.
So have you tried different frequencies?
So I've not looked at different frequencies because we came at this looking at trying to do sort of quasi-physiological loading, right? And so we've picked what was for the mouse a physiological frequency, and so I've not been interested in going, and there are groups that do a lot of frequency work. So Clint Rubin's lab, for example, has done a lot looking at 70 hertz frequency and comparing higher frequencies. I guess that's not something that we've been interested in looking at. Okay. But I will say, in terms of your physiological question, you know, in the knee, 90% of OA is not PTOA. It's not trauma induced. It's happening over time, and it's probably sub-rupture trauma, so to speak. And I think
that's the other possibility is that you've got degenerative changes going on, the tissue quality gets worse, and
so I think that's what we're inducing some of, in fact. So I think we're subcritical threshold, so to speak. But we do with one. So I showed you repetitive loading here. We do see responses with a single bout of loading at 1 and 2, and actually we now just are busy collecting data for 6 weeks. And so with a single bout of loading, we are seeing differences here, in fact. So, but we're not getting. We don't have rupture of ligaments. We're not causing that type of damage.
But you can get a single insult, like I do that. That's an insult to the joint, and if I do it, I'm going running, but it's not that high. And I must admit, I don't know what the articular loading is in the mouse joint, and the range of freedom, as Tor said, and the articulation of the joint is going to change the fluid flow in the cartilage.
Yes. Yep.
And that is protective too. So it's nice work, but
it's a complex question.
It is.
Again, we're focusing on a particular aspect of it, which I fully realize and fully admit. But it's very hard to do all of it.
Yeah, it is. Yeah.
So
it's lovely. It's great.
And again, to me, it's interesting just because I came at this very much on something that's anabolic, we're positive for bone, and then we didn't realize that at the same time we were creating such damage in the joint, right? And there's still a lot of people looking at the bone loading question, but stepping back and saying what is going on further in this system, I think is really important, and we seldom do. I mean, if you go look at a lot of the cartilage cartilage work in osteoarthritis, they haven't looked at the underlying bone necessarily, or there's a sort of a single, it's end-stage sclerotic. But I actually also think that, what in OA, what we're seeing actually is initially a loss of bone, and that's contributing to, and then you get this sclerosis after that. But most of what we see in humans is end stage. And so the value of these models is that we can look at this early stage, and that doesn't exist in human
do
kids.
you see sorry,
Yes.
have we got time?
Yeah, we do, yeah.
Do you see loss of the cancellous bone in this model?
In the epiphysis, it's actually subchondral. There's very little cancellous bone in the epiphysis in the mouse, but we see a dip, and then it comes up, and then it gets thick. Then we have the sclerosis.
And then
at
you get the
longer
cortical
time
bone
points.
coming in and trying to stiffen up again.
Yes, it stiffens up in the longer time points. So we do
Which
see
is
that
what you
progression.
see in ACL injuries too.
So yeah, no, so I think there's aspect, but again, I think every single model, the other model that gets used a lot is this monoiodine acid injection, right? So the sort of very destructive OA, and that has its own limitations, right? So I think you can learn pieces from each model, as I said earlier, in terms of understanding OA. You have to understand which aspects of the human pathophysiology are captured or not. And I think capturing all of them in a mouse will never be possible, but I think there's aspects that are valuable. And that's also why we characterize it, right? That's why I want to know what was the kinematics we're doing, and we need to figure out what, and we're working right now on tissue stiffness. What are the changes we're causing in the tissue, and how much do they match? But in early OA, the data doesn't exist for humans, so that's the other thing that's unique here as well. So understanding how these different models, there's many people looking at these mouse genetic models and understanding what's common between them and what's different, because I think that'll give us some ideas also in terms of where the variables sit and where the commonalities will give some insights as well.
Thanks.
I have one really quick question.
Yep, go Brent, please.
I teach a graduate course, and we talk about the microdamage hypothesis in subchondral bone leading to subchondral bone sclerosis and marching of the tide mark and cartilage degradation. So I'm just wondering, in light of this new knockout model that you have that kind of shows that increasing the compliance of the subchondral bone actually leads to cartilage damage. If I should maybe stop teaching the subchondral bone hypothesis to my students.
Well, so one thing I didn't say, so we determine our loads based on doing strain gauging the mid-diaphysis of the tibia. And so to go back to what's physiological or not, in animals, so our mid-diaphyseal strains are 1,000 to 1,200 microstrain in our loading model. So when we do 9 newtons, if we go to a different genotype animal, we strain gauge them and we figure out what the applied load is to create a 9 Newton load for the bone work at least. So in the cartilage work, I'm not sure what the right driving parameter is right now. We've sort of been using a constant load level. But so we strain gauge them, we figure out, and we apply the load to match that, to get and bone, damage, so if you look at Cheryl Patton's work, for example, it's around 2,500 microstrain, 2,000 and above, right? At 2,500, she has a big change in her fatigue behavior of bone. So I think that we're in the physiological range for what we can measure at the cortex. And now we're only measuring at one location, and I realize that may or may not be fully representative. In the mouse, you can't put multiple gauges on. You're going to totally stiffen the bone and change the behavior. There's a lot of people that are doing models, but they have their own limitations. So doing a full model of the tibia, putting your loads on and seeing, but those are all constant material property models, right? And I know I don't have that here. So we've taken the experimental approach for matching our strains. I don't think we're creating, I don't think our damage mechanism is micro damage we've looked a little bit early on in our bone work. But I don't know, I don't know that a more compliant bone can't with loading create damage. So I want to be careful saying that, in fact. And in talking to Steve and Mary Goldring, the whole cartilage field talks about how the increased stiffening of bone is what's the subchondral bone sclerosis is a stiffening, and that's causing the OA. And I agree. I think it's the opposite. I think you have it's actually a huge stiffness mismatch in the models, right? So when we model this, we can vary the bone properties plus or minus 50%, and it doesn't affect much because it's so stiff compared to the cartilage. You modulate the cartilage properties, you get massive changes in the stresses that are induced with loading here. But at the same time, our experimental data, with one exception across many, many experiments, we see a correlation between the subchondral bone mass of the contralateral limb, which we're using as indicative of our starting point, and the damage score, with lower subchondral bone mass creating higher damage. So I very much think that that's part of this equation in driving this OA development. But I can't fully demonstrate it right now, but that's my working hypothesis. And we started with the opposite hypothesis if you go look at our grants from some time ago, but now the data has shown us the opposite.
Okay, great. Well, that's one more question. Quick.
Hi, Anne Munderman, University of Basel. I find it very intriguing that your mice get the weekend off. I would like to compliment
the graduate
you.
students and the PI too.
I assumed that. A great talk, very inspiring. I have a question regarding your setup, because you have a quite high flexion angle at the knee when you apply your loading, and I think it's more flexed than it would be during normal ambulation. So that would actually go along with some of the hypotheses that, for instance, in ACL injury, the areas of cartilage are loaded that were previously unloaded, and they're not conditioned to actually take up the load. My question...
So we've reduced our flexion angle, actually.
Okay.
We are less flexed than most other people that do this work these days.
No, but how does it relate to ambulation?
Excuse me?
It's still more flexed than during ambulation, isn't it?
I don't think it is, actually.
Okay. But my other question is regarding the early biomarkers. I'm very interested in load-induced changes in biomarkers in humans. Mm-hmm. And I think it would be great if you would actually look at some of these biomarkers, both systemically or in synovial fluid, to give us some insight into what's actually happening
I would love to look at mouse synovial fluid. There's just not a lot there to look at.
How about, but you can take
some
systemic concentrations as well, right?
Yes, we have not done that.
Because that would give us a lot of information on what's missing in humans.
That's a great point. Yes, we have not done that. But yeah, we should.
Thank you.
Yeah, thank you.
Okay, let's thank our speaker.